We have started lecture 1 in the previous session, and today we are going to continue. In the previous session, under the previous lecture video, I have inserted a caption and explained about DNA and gene again. Maybe it's not a bad idea. Before continuing with that, I will talk about it. So let's go to that slide so that I can mention the thing I said in the caption. Yeah. In the previous session, we mentioned that gene actually, and when we want to compare it to DNA, DNA is a subset of gene because we have RNA and DNA inside gene, but DNA consists of nucleotides, these letters. But also when you look at the sequence, somehow we can say gene is a subset of DNA as well, because there are DNAs which we can say they are non-coding, and that's why when I talked about junk, I should rephrase that and I say junk DNA are the part of DNA which they are not gene. So all of the known genes that we know, they are responsible for synthesizing proteins, and those unknown genes that we don't know, for the time being, we call them junk DNA because they are inside DNA, and we don't consider them as genes who are responsible for producing protein. That's why I wanted to rephrase that to make it correct. And also let me show you in the DNA sequence that what I mean by that. For example, we have such a DNA sequence, and maybe just a part of it we can consider as gene. Like, for example, third sentence and fourth sentence. Maybe these two sentences, or just maybe a part of this sequence, we can consider as gene, and that part will be transcribed and then translated into protein at the end. So that's why in that sense we can say yes, gene is a subset of DNA. So I wanted to mention that. And also, while we are on this slide, again, I repeated that each one of these codons, triplets of nucleotides, they are corresponding to a specific amino acid. So we can show them by just a letter. Sometimes they show it by 3 letters, but with just a letter in a sequence, it makes more sense. That's why they have 20 number of these letters, not as the number of alphabets. And some of them you can see they are corresponding to a stop codon. That's the place that you stop producing protein from that part. And in one picture, I actually showed it maybe after that, but we can also talk about it later. All right, but today I want to talk about the databases that we can use in order to find these data. And whenever we want to find the sequence of the protein, this database is perfect for that. And for example, when you search a specific protein, that's what you get, like the sequence of amino acids. So it consists of 145 amino acids. For example, the number you can see here, 10, it corresponds to the last letter here to show just the position. That's it. So 1, 2, 3 until 10. So these are the amino acids, and together they are creating a protein. So this database is very good for that, to find these sequences. Of course, if you change any letter with another, it might change the whole protein. That's why the order matters. Whenever you fix one letter, and from that part, you shouldn't just try to switch the letters together. It's not possible. Later, I will explain why this is important, especially when we will do the sequence alignment. Also, we have a protein data bank. I also mentioned about the URL, and you can just find the protein structures here. Not just that, there are some other information for you too, but for finding the protein structure, this database is perfect. There are all the proteins that we know. It's not possible to find the structure easily. You know, they could cover some proteins, some limited proteins, and if you look at the database, you can understand exactly how many proteins so far they have covered. It's not an easy task to know exactly about structure of a protein, and structure of proteins is very important because not only sequence will give us some information, also the structure, the similarity between structures, it is also meaningful. And we will talk about it in details for sure. So far we have introduced two important databases. This one, of course, the complete URL should contain other elements as well. You know that. I just shorten it, and you know how to browse it on your internet. So this is the structure sample that you can get from that protein data bank, such as structure. It contains some subunits, and as you can see in the picture, we can see some information that we cannot see inside the sequence, about especially when we talk about subunits. Maybe each one of these subunits contain a specific sequence. For example, for hemoglobin, we have alpha globin chains and beta globin chains in a human adult. And for different proteins, we can have different subunits. And the shape of it and the way it can be binded to other proteins or other molecules, this is important to know what's the structure of a protein. This part is very important as well because we are going to more details about each gene. You see, here we show this is a DNA strand. There are some nucleotides that we are not interested at this moment. Maybe in the future, scientists understand those codes are meaningful too. But for the time being, this part of the DNA, we call it gene because this gene is responsible for synthesizing a protein at the end. So first thing happening here, we are trying to transcribe DNA into mRNA. What happens here? Gene contains exons and introns. You see, this one contains 4 exons, and in the middle of them we have introns. And when we transcribe one gene to an mRNA, in this case we are getting the information from exons only, not introns. You see, exons are coming together to have this information inside mRNA, and we call it transcription. So you see, introns are not included in that. You may ask, do we care about the nucleotides, about those 4 letters that we have here? When I say letters, I explain it. In computer science, we call them letters, but in fact, they are chemicals. Okay, when we have those 4 letters, the sequence of them here as introns, they are important as well because when there is a mutation on our gene, when mutation happens, for example, one changes, and we don't reach the stop codon. You remember when we have stop codon, we should stop getting information from DNA to mRNA, and we stop there. And why? For example, it not totally stop here, but totally stops here. Yeah, that's a good question. Because you know these exons are coming together. It actually stopped, but it comes together. For scientists to understand that this is happening, it took lots of time to discover these things, that these exons are related together in the same strand. So for the time being, you should know that exons will be transcribed to mRNA, and introns are in the middle of them. They have important codes because of the mutation. Maybe some part of the intron by accident would be considered inside the exons and will be inside this mRNA by mistake. You see, that's why the role of introns are important. And sometimes the regularity factors will decide that this stop codon, for example, is the one that we should totally terminate creating this mRNA, and that's why the information inside introns are important too. So again, to sum up, gene includes exons and introns, and for different genes, we have different number of introns and exons. For example, for beta globin gene, we have 3 exons and 2 introns. We have a term named alternative splicing. This term is also very important in molecular biology. When we have a DNA, as we said, sometimes we call it DNA, but as a part of it, we are just showing the gene, right? It's very similar to numbers. For example, when we say rational numbers and natural numbers, one of them is the subset of the other one, right? But when we say, for example, number 5 is a rational number too. So when we call it DNA, it's not a mistake. This is a DNA, okay? But of course, in the specific case, this is a gene. So here, again, we are transcribing these 4 exons from the gene. Here you see we have mRNA based on those exons. Actually, in molecular biology, they say we transcribe the whole thing, including the introns, but you know that in mRNA, we don't have the information about introns anymore. Now, the thing about alternative splicing is that now you have the combination of them to synthesize different proteins. You see, when we have, for example, here we have dark blue, then green, then red, and so on. You see, we have this protein. If we change the place of these exons, you will get totally different proteins. And this is called alternative splicing. And the order totally matters. So we should be careful about the order of exons, and notice that the same gene is able to synthesize different proteins, which is very interesting. And it happens through alternative splicing. Genome, we talked about it briefly in the last session, but genome corresponds to all collection of genes that you have in an organism. For example, as a human body, a human organism, we know that all of us would have similar genome to make us human beings. There are some differences which reflect to our appearance about the function of our organism, of course, but to come up with such a genome to describe the whole organism in just one picture, as you can see here, it takes lots of time. It's not an easy task to do. Scientists couldn't find all genomes for all organisms in the world. It takes time, and they are trying to cover as much as they can. There are big projects for finding the genome. And here in this picture, we have this circular format, which is not related to human beings. For human beings, this is linear. It's not like a circle. And that's why when we talk about genome, just remember this is very vast, and it's related to collection of all our genes inside one. Now for genomes and finding also some useful sequences, you can use this website, NCBI Database. This database is very famous. This is the favorite of many molecular biologists, and this one for sure, they will use it a lot. Bioinformaticians, they use it a lot as well. We will talk about the details later, also about the submenu inside this database. And there's another term, pathway. In molecular biology, we hear about pathways a lot. So far, we talked about transcription, translation, central dogma of biology, how we can reach different proteins. And we need to know how these proteins And even genes with proteins, different mRNAs with proteins, they are interacting with one which actually very complex system, and how they are interacting with one another. So it's very important to know about their interactions, like how we can come up with this complex proteins. So different proteins are coming together, and you see just one molecule maybe binds to them. There are many interactions. Even we can include central dogma of biology inside this system as well. And you see, when we say DNA damage, we mean DNA mutation. So such things can be included inside the pathway. So it's a kind of system, as you can see. And these pathways are important in molecular biology to say, for example, what's the mechanism in order to come up with such a complex multiproteins, or maybe such complex multiprotein can be resulted into a DNA repairment. And to do that, for example, we can increase or decrease the level of these proteins. And whenever we want to simulate in computer softwares, for example, I personally use Petri nets in order to do that. And these pathways can be very useful for computer scientists. It gives us lots of ideas that's what's going on. And in order to find these pathways, KEGG is the best database to find them. For example, if I want to have a research on hemoglobin, for example, hemoglobin switching pathway, if I'm interested in that, then I can search here and find the corresponding pathway. So this one can be very helpful. If I know, for example, I can fix the problem which caused by a mutation here by just decreasing, for example, the level of this one, then computer simulations would be very useful, because as you know, it's very costly to run such experiments on, for example, animals, on human beings, and it takes also time. But when you simulate something, you just click and it just simulates very fast. You can predict things, which is the beauty of computational methods. That's why if we have such pathways, if we can model them by using computer science, mathematical modeling, then it would be perfect for finding treatments. Just at least, I would say, to be accurate, identifying prediction, because many things maybe you predict and it sounds perfect on your computer simulation, but in reality, it could be totally different. And that's the thing usually molecular biologists don't like about computer science, because they say you find something which is not real. And later we will talk about our algorithms in computer science, that when you come up with them, you neglect many things. This is true. We have so many errors, and we are trying to find the best algorithm for defining what's going on inside the nature. But at least we are shed light on the matter anyway. There are many things that molecular biologists couldn't deal with because of maybe they were costly, maybe because they didn't see it. But by using these computer simulations, by using these pathways to come up with your own models, then of course we have discovered so far many things that molecular biologists couldn't do alone. And that's why we need both molecular biologists and computer scientists together in order to make this project happen. Because as computer scientists, we may make many mistakes because we don't know exactly about the nature of these pathways, because we are computer scientists, we are not molecular biologists. So in big projects, we need each other to make it happen. So these pathways, as I mentioned, they are important, and you can find them in KEGG pathway. There are some important notes I want to explain here. By the way, I added chapters 2 and 3 because now you can read them as well. chapter 1 was good enough for the previous session, but now you can cover until chapter 3. Sometimes you may say it takes lots of time to cover all of them, to read all of them. You know, the terms that we are using in the lecture is the most important ones. So if you see, for example, the terms which are complex for you or we didn't mention at all inside our slides, it's nice to read them, but don't just linger and try to understand everything about the molecular biology. You can just skip them for the time being, but terms like DNA, mRNA, protein, gene expression, like, for example, transcription, translation, the things that we mentioned in the lecture, they are important. Please, when you read these chapters, when you see these terms, try to understand them and don't just skip those terms. And I want to also mention that the PowerPoints actually are designed the way to be used in the online session. It's not the kind of summarizing for you for the final exam or midterm exam. Because some students tend to do that and they try to use it to learn things just before exam. It won't work. We need to read these chapters to understand them, and sometimes I will share with you extra reading materials if necessary, sometimes animation of videos. For the last lecture, I didn't share with you because the animation I found could be misleading for you. And they had some terms which are not necessary for us to know. But still, if you search, for example, transcription or translation, or, for example, gene expression, there are some interesting animation for you that you get ideas. But I didn't want to share myself because it could be misleading in some terms, and they are talking about too much molecular biology that we don't know. We don't want to know that much about them at this stage. Later, one by one, we will cover them all. So that was all for this session. Now you know famous databases that you can use for molecular biology. Again, we will introduce you more through our lectures, especially about the sequence alignment that we will talk about next week. And as soon as we will have our assignments start from the 3rd week, it means it will be 2 weeks later, then you will see that what we are looking for to ask you in order to evaluate you in the exam as well. So don't worry about it now. Just go through the book chapters and try to cover them as much as you can. And if you have any questions, please ask me now or send me an email and ask your questions.